The chemiosmotic theory. It was proposed by Peter Mitchell in 1961. In his proposal, he hypothesizes that proton motive force was responsible for driving the synthesis of ATP, and he said that this was done by energy coupling. He believed that this was partly due to the flow of protons passively back into the matrix through a proton port ATP synthase. This is reflected by the equation. In this model of the chemiosmotic theory, electrons from NADH, succinate, and oxygen pass through a chain of carriers arranged in the inner membrane. This flow of electrons is met with proton transfer across the membrane. This produces a chemical gradient and an electrical gradient, which combined create the proton motive force. Thanks to the inner mitochondrial membrane being impermeable to protons, protons can only re-enter the matrix through the proton-specific channels, like the F0 unit of the ATP synthase. So how does coupling work? In experiments to demonstrate coupling, mitochondria were suspended in a buffered medium in an O2 electrode that monitored O2 consumption. At intervals, samples are removed and assayed for the presence of ATP. Addition of ADP in organic phosphate alone did not increase either respiration or ATP synthesis. However, when succinate was added, respiration began immediately and ATP was synthesized. Adding succinate alone did not induce respiration. They were both needed, ADP, PI, and succinate, in order for synthesis to work. Cyanide was added, which showed inhibition. Oxidation can be uncoupled from phosphorylation. When intact mitochondria are treated with a detergent that introduces fragments in the membrane, the mitochondria can still catalyze electron transfer from succinate, but lack the membrane means that protons are not able to re-enter via ATP synthase, so ATP synthesis doesn't work. But there are two main ways that are directly affected: DNP and DCPP. Another way is introduction of weak acids that have hydrophobic properties. This permits them to diffuse readily across the mitochondrial membrane. After entering the matrix in the pronated form, they will release a proton, which will dissipate the proton gradient. The chemiosmotic theory predicts that because the role of electron transfer in ATP synthesis is inherently pump proteins to create the proton motive force, an artificially created proton gradient should be able to replace electron transfer in driving ATP synthesis. This is true and experimentally confirmed. Mitochondria manipulated to create a difference in proton concentration and separation of charge with an oxidizable substrate still synthesize ATP. The proton motive force can drive ATP synthesis. This concludes this presentation.